Right now, your body's up to something pretty amazing, whether you notice it or not. Your gut microbiome, a hidden world made of trillions of bacteria, is always busy, chatting with your immune system, your brain, the stuff that controls inflammation, and even the clock ticking on how you age. All of that just ticks along, quietly, every moment. When things are going right down there, inflammation stays low. Your gut lining holds strong. You feel steady, your energy's reliable, and you don't seem to age so fast. But honestly, the way most people eat nowadays pretty much throws that whole balance out the window. Inflammation starts to creep in, often without any clear signals. The gut lining gets a little leaky. Immune responses go haywire. And even if you can't feel it, those deep-down cells of yours start aging in fast forward. Now here's the part hardly anyone talks about. There's this one bacterium called Akkermansia muciniphila that scientists see as a real VIP when it comes to aging well. Problem is, if you're eating like most folks today, you're probably losing it. Its numbers just keep dropping off year after year. Funny thing, though, ancient cultures found ways to protect this bacterium without ever hearing its name or knowing it existed. They just lived in a way that supported it. No fancy knowledge needed. And in the end, science isn't so much discovering the gut microbiome. It's really just circling back to what people quietly practiced for centuries. Imagine a forest, not a forest of trees, but a forest inside you. Dense, living, interdependent, thousands of species, each playing a role the others depend on. Some producing compounds that protect the gut lining. Some signaling the immune system to stand down. Some manufacturing vitamins your body cannot produce on its own. Some generating the molecules that travel up the vagus nerve and shape how calm, how focused, how mentally clear you feel. This is the gut microbiome. And its diversity, the number and range of species living within it, is now understood to be one of the most consequential predictors of how well and how long the human body functions. But the strangest part of this story is not the science. It is the timing. When the forest is rich and varied, the body does what it was designed to do. Inflammation stays low. The gut lining stays intact. That single cell-thick barrier between your digestive tract and your bloodstream holding firm. Immune responses remain proportionate. The biological clock turns at its intended pace. When the forest depletes, when microbiome diversity collapses, when beneficial bacteria lose ground to harmful species, the cascade begins. Inflammation rises. The gut lining becomes permeable. Bacterial fragments cross into the bloodstream. The immune system detects the breach. And it responds not with a dramatic acute reaction, but with a slow, chronic, grinding inflammatory signal that researchers have given a name: inflammaging, silent, systemic, correlated with nearly every major chronic disease pattern of the modern world. Scientists call this state dysbiosis. What they have not fully answered, until recently, is when it began, and what, in the ancient world, was quietly preventing it. The answer reaches further back than anyone expected. In the basement of the Salk Institute in La Jolla, California, a team of researchers spent years trying to understand a single question. Why did the gut bacteria of modern Western populations look so fundamentally different from those of populations still living close to the land? What they found was not a minor variation. It was a structural difference in diversity, in population composition, in the functional outputs of the microbiome itself. The living ecosystem inside the modern gut had changed in ways that took decades of industrialized eating to produce. What happened next surprised researchers working in a completely different field. At MD Anderson Cancer Center in Houston, oncology teams studying treatment outcomes began noticing something they had not designed their studies to find. Patients with richer, more diverse gut microbiomes, more populated with beneficial gut bacteria, were responding to measurably differently from those whose microbiomes were depleted. The hidden healing system inside the gut was influencing outcomes in oncology wards, far removed from any gastroenterology conversation. At the University of California, San Diego, a team mapping what they called the human microbiome aging clock made a finding that reframed what longevity research even means. Gut microbiome diversity in adults over 65 was not merely correlated with better health markers. It was correlated with how long those people were likely to live. The gut was not a bystander in the aging process. It was an active participant, one that modern medicine had been largely ignoring. And this is where the story becomes difficult to ignore. Researchers at the APC Microbiome Institute in Cork, Ireland, one of the world's leading microbiome centers, published a body of work connecting chronic low-grade gut inflammation with outcomes ranging from cardiovascular disease and metabolic dysfunction to cognitive decline and depression. The gut microbiome was not managing digestion. It was governing the inflammatory architecture of the entire body. 4 institutions, 4 research directions. Every line of inquiry converging on the same conclusion. The gut microbiome is not a digestive accessory. It is a central operating system for human health and longevity. And then, almost as a footnote to the science, the researchers began noticing something that made them pause. The foods that most consistently supported a healthy, diverse microbiome in their studies were not novel. They were not new discoveries. They were not patented compounds or pharmaceutical innovations. They were ancient: garlic, onions, leeks, bitter herbs, legumes, fermented foods, pomegranate, figs, barley. The same foods carried across biblical trade routes for thousands of years. The same foods ancient populations ate not because of nutritional science, but because the body had always known what it needed. And generation after generation had listened. The human gut lining is, at microscopic scale, one of the most quietly extraordinary structures in biology. A single layer of cells, thin enough that if spread flat it would cover a tennis court, stands between the contents of your digestive tract and your bloodstream. This is not a metaphor. It is a literal single-cell barrier, and protecting it is a living film of mucus, maintained by a community of bacteria whose sole biological purpose is to keep it intact. The most critical of those bacteria is Akkermansia muciniphila. What researchers discovered about Akkermansia changed how longevity science understands aging. Formally identified only in 2004, Akkermansia has become one of the most intensively studied bacteria in the field. Higher Akkermansia muciniphila populations in the gut are consistently associated with lower systemic inflammation, stronger metabolic function, reduced cardiovascular risk, and longer health span. It is not one beneficial bacterium among many. In current research, it functions as a keystone species, one whose presence determines the health of the entire mucosal ecosystem around it. When Akkermansia is depleted, as it is in the majority of people eating modern industrialized diets, the mucosal shield thins. The gut lining loses structural integrity. Bacterial fragments, dietary antigens, and inflammatory compounds begin crossing into the bloodstream. The immune system detects the breach and responds, not acutely, not visibly, but persistently, with the low-grade inflammatory signaling that researchers now call inflammaging, the silent fire of modern chronic illness. Its origin, in millions of people, is a thinning gut lining, and the bacteria responsible for maintaining that lining have been retreating for decades. But here is what makes the ancient connection impossible to dismiss. What feeds Akkermansia muciniphila most selectively are polyphenols, specifically the polyphenols in pomegranate, green tea, dark berries, and walnuts. And among these, pomegranate is the most studied. Pomegranate appears in Scripture as a symbol of abundance, restoration, and sacred provision. It decorated the temple in Jerusalem. It was woven into the high priest's robe. Ancient people revered it in ways that seemed, until recently, purely ceremonial. But the polyphenols in pomegranate, specifically a compound called punicalagin, have been shown in recent research to selectively increase Akkermansia populations in the human gut. The mucosal shield was always being protected. Ancient people simply did not have a name for what they were protecting it with. There is a process happening in your large intestine right now that has been happening inside human beings since before recorded history. Prebiotic fiber, the indigestible structural material in plants, survives the acid of the stomach and the enzymes of the small intestine. It arrives in the large intestine intact. And when it does, your beneficial gut bacteria do something that determines, in ways researchers are still mapping, the biological age of your body. They ferment it. And the products of that fermentation may be among the most consequential molecules in human health. Short-chain fatty acids, primarily butyrate, propionate, and acetate, are the outputs of microbial fermentation, and each one does something distinct and critical. Butyrate is the primary fuel source for colonocytes, the cells lining the colon wall. Without a consistent butyrate supply, those cells begin to malfunction. The gut lining degrades. Inflammatory signaling increases. The mucosal architecture the body depends on begins losing its structural coherence. Propionate travels to the liver, where it modulates glucose metabolism and lipid production, influencing the metabolic processes that determine weight, blood sugar regulation, and cardiovascular risk. Acetate enters the bloodstream and communicates directly with the immune system, calibrating inflammatory responses across the entire body. The fermentation engine is not a side process in human biology. It is a central biological mechanism, and it runs entirely on what you feed it. This is where the difference between ancient and modern diets becomes most visible and most consequential. In ancient diets, prebiotic fiber arrived continuously and in extraordinary variety. Garlic and its fructo-oligosaccharides, barley and its beta-glucan, leeks and onions and their inulin chains, legumes and their resistant starch. Each fiber type feeding a different bacterial population, each population producing distinct fermentation outputs. A diverse engine generating a rich, varied short-chain fatty acid supply, fueling the gut lining, modulating the immune system, slowing the metabolic aging process simultaneously. The Sonnenberg Laboratory at Stanford University spent years studying precisely this relationship. Dietary fiber diversity and short-chain fatty acid production in the human gut. Their finding: fiber variety matters as much as fiber quantity. The number of different plant species consumed in a week is one of the most meaningful predictors of gut microbiome health currently identified. Ancient people ate that variety not by scientific design, but by seasonal necessity. The ancient world rotated its plant supply by geography and season. The microbiome received different inputs through the year. The fermentation engine ran rich and varied. The gut lining stayed thick. Inflammation stayed low. Modern people eat the same 12 foods on the same rotation every week of the year. The fermentation engine receives the same narrow inputs repeatedly. And slowly, without announcing itself, its output declines. The gut microbiome is not a digestive system that happens to interact with immunity. It is, first and foremost, an immune training ground. Approximately 70% of the body's immune tissue resides in the gut wall. This is not a design flaw or a coincidence of anatomy. It reflects a biological arrangement hundreds of millions of years old. The immune system positioned at the boundary between the external world and the internal body, where it is educated continuously by the microbial community living in the intestinal space. What researchers did not fully appreciate until recently is how much of immune function depends on this education being ongoing. Beneficial gut bacteria train the immune system to distinguish between genuine threats and non-threats. They produce compounds that signal immune cells to stand down after a response has completed its purpose. They calibrate the immune system's threshold, determining how sensitively it fires, how long it stays activated, and how completely it resolves. This calibration cannot be replicated by any pharmaceutical currently in existence. When the microbiome is diverse and the education is continuous, the immune system is proportionate. Inflammation rises to meet a genuine threat and resolves when the threat has passed. The biological systems that depend on that resolution, cellular repair, hormonal regulation, cognitive clarity, energy production, operate as designed. When the microbiome is depleted, the calibration fails. The immune system becomes either chronically overreactive, generating the low-grade systemic inflammation associated with autoimmunity, metabolic disease, and accelerated biological aging, or underreactive, failing to mount adequate responses when genuinely needed. This may explain why modern rates of chronic inflammatory disease have risen so steeply, even as acute infection rates have fallen. Ancient fermented foods were, in a precise biological sense, continuous microbiome inoculations. Yogurt, kefir, fermented vegetables, wine from whole grapes, vinegar prepared from fermented fruit, not consumed as health supplements, consumed as food, as flavor, as preservation technology, but delivering live microbial communities to the gut at every meal, reinstating the beneficial bacteria populations that kept the immune calibration signal running correctly. Stanford researchers published findings in Cell in 2021, confirming that a high fermented food diet significantly increased microbiome diversity and measurably decreased inflammatory markers, with effects detectable within 10 weeks. Ancient populations had been achieving that same effect for 10,000 years, without knowing the mechanism. Simply by continuing the practice. The gut microbiome is not a digestive system that happens to interact with immunity. It is, first and foremost, an immune training ground. Approximately 70% of the body's immune tissue resides in the gut wall. This is not a design flaw or a coincidence of anatomy. It reflects a biological arrangement hundreds of millions of years old. The immune system positioned at the boundary between the external world and the internal body, where it is educated continuously by the microbial community living in the intestinal space. What researchers did not fully appreciate until recently is how much of immune function depends on this education being ongoing. Beneficial gut bacteria train the immune system to distinguish between genuine threats and non-threats. They produce compounds that signal immune cells to stand down after a response has completed its purpose. They calibrate the immune system's threshold, determining how sensitively it fires, how long it stays activated, and how completely it resolves. This calibration cannot be replicated by any pharmaceutical currently in existence. When the microbiome is diverse and the education is continuous, the immune system is proportionate. Inflammation rises to meet a genuine threat and resolves when the threat has passed. The biological systems that depend on that resolution, cellular repair, hormonal regulation, cognitive clarity, energy production, operate as designed. When the microbiome is depleted, the calibration fails. The immune system becomes either chronically overreactive, generating the low-grade systemic inflammation associated with autoimmunity, metabolic disease, and accelerated biological aging, or underreactive, failing to mount adequate responses when genuinely needed. This may explain why modern rates of chronic inflammatory disease have risen so steeply even as acute infection rates have fallen. Ancient fermented foods were, in a precise biological sense, continuous microbiome inoculations. Yogurt, kefir, fermented vegetables, wine from whole grapes, vinegar prepared from fermented fruit. Not consumed as health supplements, consumed as food, as flavor, as preservation technology, but delivering live microbial communities to the gut at every meal, reinstating the beneficial bacteria populations that kept the immune calibration signal running correctly. Stanford researchers published findings in Cell in 2021, confirming that a high fermented food diet significantly increased microbiome diversity and measurably decreased inflammatory markers, with effects detectable within 10 weeks. Ancient populations had been achieving that same effect for 10,000 years, without knowing the mechanism, simply by continuing the practice. There is a kind of grief that comes with understanding what the modern diet has done to the gut microbiome. Not anger, not conspiracy, just a quiet, honest recognition that something essential has been lost, and that the loss accumulated so gradually, nobody noticed until the research made it impossible to ignore. Ultra-processed foods, engineered for palatability rather than nourishment, carry almost no prebiotic fiber. The fermentation engine receives nothing to work with. Short-chain fatty acid production declines. The gut lining begins losing the butyrate supply it runs on. Akkermansia muciniphila retreats from the mucosal surface it was designed to protect. The numbers here are difficult to sit with. The average adult in the Western world now consumes fewer than 15 different plant species per week. Ancient diets routinely included between 50 and 150. That is not a dietary preference gap. It is a microbiome biodiversity collapse, with measurable consequences for inflammation, metabolic health, and the pace of biological aging. Antibiotic use, necessary and life-saving in acute situations, carries a documented impact on gut microbiome composition. The research is careful to note this is not an argument against antibiotics. It is an argument for understanding the ecological cost and actively restoring what was disrupted after treatment. Constant snacking elevates insulin continuously and denies the gut the rest periods, during which certain bacterial populations replenish and the mucosal lining consolidates. The ancient rhythm of eating, feasting followed by natural overnight fasting, was not only metabolically valuable, it was microbiologically necessary. Refined sugar feeds the wrong populations. It crowds out the beneficial bacteria that govern everything else. And it does so silently, without a single symptom announcing what is changing beneath the surface. The modern world did not design this damage, but the evidence now makes clear it happened. Restoration is not complicated, but it requires understanding what the microbiome actually needs, and giving it that rather than a pharmaceutical approximation of it. It needs fiber diversity, not a single prebiotic powder, not a probiotic capsule as a substitute for food. Diverse, rotating plant fibers from different botanical families, feeding different bacterial populations simultaneously, keeping the fermentation engine producing the full range of short-chain fatty acids the body runs on. Start with what ancient populations ate most consistently. Rolled oats, 3 grams of beta-glucan per serving, selectively feeding Akkermansia and bifidobacterium populations. Garlic, raw where tolerable, 1 to 2 cloves daily. The fructo-oligosaccharides feeding lactobacillus strains more efficiently than most commercial prebiotic products. Leeks and onions several times a week, with some raw preparation retained for maximum prebiotic integrity. Lentils and legumes 3 to 4 times a week. Resistant starch arriving intact in the large intestine as direct fermentation substrate. Pomegranate, half a fresh fruit or a small daily quantity of pure pomegranate juice without added sugar. The punicollagen concentration is highest in this unprocessed form. Green tea, 2 to 3 cups daily. Catechin polyphenols selectively support beneficial bacterial populations while modulating the inflammatory signaling that chronic gut imbalance generates. Walnuts, a small handful each day. The polyphenol content combined with prebiotic fiber and omega-3 fatty acids creates a synergistic microbiome effect that multiple independent research groups have now documented. Fermented foods, genuinely fermented. Natural yogurt with live cultures. Kefir, raw unpasteurized sauerkraut or kimchi. Not shelf-stale pasteurized versions stripped of their living cultures. Even a small daily portion shifts the microbial landscape measurably within weeks. And overnight fasting, 12 to 14 hours between the last meal of the evening and the first of the morning. Not a trend, not a modern intervention, a restoration of the biological rhythm the gut microbiome evolved alongside. The rest period allows bacterial populations to consolidate, the mucosal lining to repair, and the immune calibration signal to reset. None of this is new. The ancient world did all of it, without not knowing why it worked. Long before laboratories could measure the gut microbiome, ancient people were already eating in ways that quietly protected it. They did not know about Akkermansia muciniphila. They did not understand short-chain fatty acids or immune calibration or mucosal integrity. They had no peer-reviewed journals, no gene sequencers, no longevity studies. They had something else. They had the accumulated wisdom of generations who had learned, through living close to the earth, that certain foods made people stronger, that certain rhythms of eating kept the body whole, that the plants growing nearest to them held something necessary, something that did not require explanation to be true. Modern science is not replacing that wisdom, it is confirming it. Finally, in the precise language of biology, Perhaps the most important question is not what ancient people knew. It is whether, in the age of processed convenience and pharmaceutical management, we still trust what the body has always known how to do. Maybe the gut microbiome never forgot how to heal. Maybe modern life just made it harder for us to remember. If something in this shifted the way you understand your own body, subscribe. Because every week on this channel, we go one layer deeper into what ancient wisdom and modern science are quietly finding together. And leave one answer in the comments. What was the one thing from today that surprised you most? I read every single one.